In the fly lab, working with DNA.
Hi, I'm Jake, a PhD student from Hidalgo Lab. For my project, I am researching how experience changes neurons and the circuits they form. When a neuron fires particularly strongly, it can respond by changing shape and growing, making more contact with its synaptic partners, in turn making it more likely to fire in the future. This could be a fundamental mechanism by which the brain adapts to change. Day to day, I study this in the fruit fly larva, the maggot, because it has neurons with large synaptic terminals connecting to the muscle. Here, these terminals at the tip of neuronal axons branch out like a tree and make multiple synaptic contacts with the muscle. These axonal terminals are plastic, and the number of branches and synapses can change with larval activity, such as crawling. I'm researching genes that could be involved in this process, aiming to understand how the proteins they encode work to regulate plastic changes in the synapses. The findings could be important for understanding how brains work, including the human brain. So, we need to have tools to study our proteins of interest. One that I'm working on today is a protein fusion. It's a copy of one of our proteins that I've genetically modified to be fused to a fluorescent protein. The idea is that when we look at the fly's brains under the microscope, we should be able to see where our proteins are, because they'll be fluorescent. They'll glow in the dark, and that's really cool. We have a few fly lines that might have the protein fusion we're looking for, and today what I'm doing is setting up a PCR to find which line is the right one. PCR amplifies a specific sequence of DNA and can be used to tell us whether or not a sample has that specific sequence in it. Today, I'm looking for the gene that encodes my fluorescent protein in DNA I've extracted from flies. The fluorescent protein originally comes from jellyfish, so it shouldn't be in the flies normally. To see the result of the PCR, we run it on an agarose gel and blast it with UV light. If one of my fly lines has the protein fusion, we'll see a band on the gel.